Welcome everybody to this month's Centers of Excellence in Hematology Meet the Experts seminar series. Today we are privileged to have Dr. Eddie Srour, who's going to speak to us in a moment. I'm going to take a moment to share my screen to point out a couple of things at the Centers of Excellence. And let me get to that screen sharing right here. So the first piece is, if I can get there, is our website, which is the Cooperative Centers of Excellence in Hematology. If you go to this website, it's very simple, CCEH.io. What you will find across this navigation bar is many important things to look at, including opportunities for jobs, including funding opportunities. We have a PNF program associated here. And you should click on this link to go look to see if that's something that might be of interest to you. There are 5 centers at the CCEHs, and this is our newest page addition, which shows you the services that are available. That is the cores at the different CCEH sites, including these different sites: Indiana, the Fred Hutch, Cincinnati, University of Utah, and Yale. So I just want to encourage you to go to that website. And in that spirit, in talking about services that are available, I want to bring your attention to a new seminar series that we're going to provide, which is an educational series, but also a series to figure out the different CCEHs. Some of them have imaging cores. And sometimes it's hard to figure out what imaging tools you should be using or what tools are available that would improve your science discovery. And so we've set up a monthly Microscope Monday seminar series, which we will post on the website as soon as we get it completed and filled out. We just have one more slot to fill, but I highly recommend you drop in there because it's going to actually teach you foundational information about microscopy, and then some of the different tools, including intravital microscopy, different confocal super resolution microscopy, long-lived long-term live cell imaging, as well as bone marrow imaging in the hematopoietic niche. So we'll post this, and we'll also put it up on social media to make everybody aware of it. But this will start in June, and we're very excited about this. So if you have imaging needs, you should pop in on this seminar series, and you'll really learn about the appropriate imaging techniques that you should be using and what cores that are available at the CCEH that might help you in this process. So if you have any questions, you're welcome to reach out to me about that. I'm going to stop sharing right now, and then I'm going to just take a moment to introduce Dr. Eddie, I'm not sure, is it Srour? How do you pronounce your last name?
You have to roll the R with the S. Try it.
Roll the R. Srour.
Srour.
Okay, it was a good attempt. Okay. Eddie comes from us originally from Lebanon, he says, a small town outside of Beirut, and where he got his bachelor's degree and master's degree at the American University of Beirut. He then went to the University of Illinois, where he obtained a Ph.D. and did a fellowship at Indiana University and has been there since 1986. He is one of the co-directors of the CCEH at Indiana, and we are so excited to have him here today to present to us on Codex technology and imaging. And with that, I'm going to turn the time over to him. I want to invite you to populate questions in the chat, and then I will invite you to ask your question, and you're welcome to populate those throughout the talk, and we will invite you during the talk to go ahead and ask those questions. So thank you again, Eddie, for being willing to present today. We're looking forward to it. I'm going to just take 1 minute to just mute individuals who are not speaking.
Thank you, Diane. Let me see if I can do this correctly. Did I share my screen?
You look good.
Okay. Okay. So what I'm going to talk to you about today is how we are trying to apply this Codex technology for the examination of the architectural relationship of some of the hematopoietic cells in the niche. And the reason why, I'll tell you in a little bit why we are focusing on the types of cells that we are trying to examine. But I want you to be aware that we are developing this not only for our own purposes, but also to make this as a shared resource in the IU CCEH, which will be in the flow and tissue cytometry core. So once it is developed and we have all the parameters worked out, you are more than welcome to come and test it yourself and apply it to your own work and do whatever you want to do with it. So I'm going to talk to you about osteoblasts, osteomacs, which are also known as endothelial macrophages or bone macrophages, and megakaryocytes and their relationship to stem cells in the niche. I won't be telling you a complete story of everything because we're still working on the niche as everyone else involved in that is doing. We don't have a complete picture, so I'll tell you what we know. And we don't have a fully workable Codex approach to our problem. So I'll tell you where we are, and hopefully we will improve with time and have a more complete story. So the whole thing started about 15 years ago when B.R. Brahmananda Reddy Shiteti came into my lab as a postdoc, and at IU, they recruited into the orthopedic surgery department.
Today, I'll tell you.
Melissa Cassina, and she hired into her lab Young Hua, and we all started to work together with the idea that we will take, which is a simplistic idea, nevertheless, that we will take as many components as we can from the hematopoietic niche, which is shown here, and it's more complicated as this. Take these components and put them in a 3D structure to form an artificial or an in vitro hematopoietic niche. So BR started to work on this. And what he did was to basically make mesenchymal or stromal cells from an adult mouse, which is very similar to what Dexter had described, which is not very similar, but identical to what had Dexter described some 50 years ago, which after 4 weeks or so becomes full of different types of cells. And he would then mix those with osteoblasts that were collected in Melissa's lab from 2-day-old pups, now shown on the right here, basically by digesting the calvaria into single cells, from which we get a 95% population of osteoblasts. And he would mix them at different ratios in a culture, and we would then charge them with sorted LSK cells and cytokines, and you can make whatever readouts you wanted to do. Now you can see that beginning here, even with just the combination of stromal cells and osteoblasts, and then the various cytokines and the various concentrations of these cytokines, this was an endless combination, so that we basically gave up on the idea even before we started. But here's what happened when, let me get, so I can see what I'm doing here. Here's what happened when we started combining these things together. We got some interesting results. So I'm showing you here the results of transplantation and primary on the left and secondary on the right recipients of cells that have been kept for 10 days, which is a long period of time in the lifespan of a hematopoietic stem cell or a reconstituting cell that has been taken out of the out of the body of a mouse. We kept them in combinations of osteoblast and stromal cells at 5% or 50-50, whichever the case may be. And then we charged them with LSK cells. But the interesting thing is that when we had OB and LSK, both in primary as well as in secondary transplants, we had an equivalent amount of reconstitution compared to fresh LSK cells. Interestingly, which we will not go into, the stromal cells had, which were here or here, for example, had a suppressive effect on the ability of OBs to propagate hematopoietic stem cells or to maintain hematopoietic stem cell function, both in primary as well as secondary transplants. I told you before, and we worked with Nadia Carlesso at the time, who was at the IU, to show that this effect is mediated through notch signaling on the stem cells and notch from the cultures. Now, Yinghua and BR continued their work, and these are the 5%. I'm going to go back to them eventually, which were not part of what we used to all the time consider as calvarial cells, as osteoblasts. So Yinghua and BR went on to basically make a hierarchical tree of these calvarial cells based on mainly the expression of CD166 and CD90. And as you can see here, by virtue of expression of various markers, going from early on top to more mature genes that are expressed by osteoblasts as they mature, we found that these cells here, which everything is color-coded, mostly CD166 positive and CD90 positive, are the immature cells, and they are responsible for the activity that you see in maintaining stem cell function. We tested those in vitro and also in transplantation studies, and I'm not going to show you all of this, but the bottom line is that the blue cells, which are those that are shown in this bar here, had the highest activity. But we always had this population of cells that every time we prepared calvarial cells, we would see it. And remember, lineage here is not the lineage that we use to identify cells in LSK. This is a combination of CD45 to exclude all the hematopoietic cells, CD31 to exclude endothelial cells, and TER119 to exclude erythrocytes. So in comes to the lab several years later, Lin, who was working on the role of CD166 in multiple myeloma, which we will not discuss today. And she wanted to find out what are these cells. So she took, instead of the lineage marker, which was, let me go back here, you can see that we are using lineage here. She basically broke down the lineage markers into CD45, CD31, and TER119. And she only found cells when she stained with CD45 against F480, which I'll tell you a little bit about in a second. She found this population of 4.5% of the cells that stain as CD45 positive, F480 positive. Now, F480 was developed in the 80s as a single marker with which you can identify bone marrow macrophages. Now there's a bit of a controversy on what F480 does, but let's not get into it. But you can identify bone marrow macrophages using F480 by itself. Again, if you put CD45, you are doubly sure that it is a hematopoietic cell. So together, they would identify this 5% of population in the calvarial cells as we isolate them. So these are fresh neonatal calvarial cells. If you grow these cells for 7 days in culture, this 4.5% population drops down and eventually dies out, and it's no longer seen in the culture. However, because of the interest that Melissa had with megakaryocytes, and she has been working on megakaryocytes and the effect of those on the bone for a long time, we would culture these cells with megakaryocytes, and sure enough, as you can see here, this population increased in size from 4.5 to 13 or 14, and sometimes to even higher numbers. By that time, Lin left the lab, and in comes another crew that was, so the work was spearheaded by Safa, who is now a postdoc at in Boston. And she was helped by Joy, who again was a postdoc in the lab, and others. And they looked at what osteomacs do and whether they differ functionally from macrophages that are present within the bone marrow of the mouse. And in order to separate the two, we had to devise some kind of a physical separation that could give us OMs on one side and macrophages on the other. And what this system was relatively simple. It was basically flushing the bone marrow to get the macrophages, and then slightly digesting the bone, the remaining bone with the way that we would use it, would digest the bone to get calvarial cells to release OMs. So what you are seeing here is a combination between 45.2 calvarial cells and 45.1 bone marrow cells, bone marrow macrophages. If we put them together, we can identify one from the other. And if you put them with megakaryocytes, only the OMs multiply and increase in number, but the macrophages don't. So physically, they are separate cell, although it is an F480 positive cell. They are physically different and functionally different than their macrophage component that is found in the bone marrow. Okay, now I'm stuck. Okay. So what Safa did, which is another functional assay here, basically transplanting, forget about the complexity of this schema here. What she found out in primary versus secondary transplants, again, is that in the primary and the secondary, when the three populations of cells, the NCC, which contains both the osteoblasts and the OMs, are present with MK, we find an activity similar to that of the LSK cells or the fresh LSK cells, even after 7 days of culture, and that this activity is not present when these cells are separated and only OBs or only OMs are cultured with the LSK, even when in the presence of MKs. So together, we went on to show, and I won't show you the details of this, that the MBGN, whether by single cell RNA analysis or by site of analysis, was increased when the cells were incubated with MK. And so we attribute this increase to both an upregulation of CD166 which I'm not getting into, and the empygen on the side of the OMs, which is due to the MK presence when present in culture. So the idea at this point was that the stem cell, which we go back to the niche, is being acted upon by a combination of factors, which we still don't know what they are completely, that are released by the osteoblasts, which are these cells that line the bone, the OMs, which are closely associated with OBs and the endosteum, and the megakaryocytes. So this is why we want to focus on these three cell types, the OBs, the OMs, and the MK, in relationship to the HSCs.
I'm Deb Snyder, and that's traffic on Hot 96.
Does anyone have a question or? Okay.
Not so far, except that I'm naive and I have lots of questions.
Okay, so the hypothesis that we came up with is that the distances between HSCs, OBs, and OMs and MKs in the young mice allow for interactions among themselves. But as the mouse ages, these cells basically walk away from each other, and those distances become larger. And so the crosstalk is more difficult, and that's why the hematopoietic activity is reduced in aging compared to young mice. Now, the entire hypothesis talks about distances. It may not be distances, it might be something else, but this is what we can at present test, and we will see if it solves any issues we have.
Eddie, can I ask a question? This is Diane Ward.
Certainly.
You're talking about distance, but it also could be that the cells, there's lower population numbers of some of those as you age.
Correct, absolutely right. We are not sure that it is the distance. It might be the number of the cells, it might be something else. It might be something that what we are trying to do does not resolve it.
Okay.
So the data would all be collected. And so we can sit down and analyze it any which way. It might not be distances, it might be numbers, but I'm not sure. I'm just trying to. So basically what we are saying is that X plus Y plus Z, which is equal to A, is a smaller number in the young versus the old mice. That's all it's a very simplistic approach, but it may not resolve the issue. So how do we go about doing this? We first tried 3D cytometry, basically tissue imaging, and the use of the FGD5 mouse. The FGD5 mouse is a reporter gene that is expressed, which is tied to GFP in our hands, that is only expressed in stem cells, as well as a sum of the CD31 positive cells. But as you can see here, we are restricted to measuring one color in every phenotype of cell that we want to measure. So the green cells that you can see here are the stem cells. They are FGD5 positive, so we take it, although we are not 100% sure that this is a stem cell. The osteomac takes one color. The osteoblasts, we didn't have a color. We were relying on the second harmonic laser imaging. So everything silver here is bone. And so the osteoblast is the endosteum, and we would measure to it, and so on. So we are restricted in the number of images that or fluorochromes that we can use. So here's a more, is this playing, by the way? Yeah, it is. So you can see that we have the bone from one end to the other, and we're focusing on this end to show you the bone marrow, the bone here, and this is a magnification of this. And you can see that we can quantify by a program that was developed at IU, the number of F480 versus GFP cells, and we can measure distances between them. But it's very, very limited with what we can do. So we basically abandoned the approach. And I've told you what the limitations are of this approach that we first took. And that's basically that we were limited in the number of fluorochromes that we could use to identify each cell. And therefore, we were not sure 100% that what we called an OM or an OB, for that matter, is indeed an OM or an OB or a stem cell. Now, the other complicating issue in the whole matter was the FGD5 mouse. In order to be able to compare young and old mice, and by old, we are talking about 20 months or older, we would have to age the mice 20 months, waiting for them to be old so we can then prepare them for analysis. So that was a restriction that, I mean, we couldn't do anything about it except to wait for the mice until they got old, and we had enough of them to analyze them. So we tried to do Codex work, which is the phenocycler. So I'm introducing you to the team that is now working hard to get this Codex. I'll call it Codex. It's easier than phenocycler, and I'm used to it. To get the Codex work done. This is mainly spearheaded by Sonali and Connor. Connor is the wizard of Codex, and he's been doing everything that needs to be done in order to get the thing to work. Sonali is a postdoc in the lab who is doing all the preparation and the analysis of the samples. Ken is the co-director of the CCEH core, that is the flow and tissue cytometry core within IU, and he knows a lot about Codex too. So what have we done, or how should we do it? Now, in order to use antibodies that apply to Codex, one has to put an addition, a melamide group on the antibody. The melamide group can then be used to bind an oligo barcode. This oligo barcode is unique to this antibody that you use, and the resulting antibody is now bound to an oligo that is unique to it, so that you have a unique antibody that recognizes, let's say, CD4, bound to a unique oligo that is now a complex. This complex is then developed by the addition of another oligo, which binds to the first one, carrying a fluorochrome. So when you do the analysis, in a nutshell, this is what you do. You basically put all of your antibodies of interest and on the tissue section at the same time. You add a certain amount, a certain type of oligos that will bind to the first set of oligos on the antibodies, and you develop them with some kind of a fluorochrome. You take an image. You then use the Codex, which is a patented buffer, to remove the oligos from the first set of oligos. You then apply another set. Now, these oligos would bind to a different set of antibodies. You develop them, and you take an image, and so on. You can repeat this up to 40, up to several cycles, and put together the compilation of about 40 antibodies that are stained with 3 or 4 fluorochromes. You don't use 40 of them, you only use 3. But since the coordinate of your slide is always in the same position, you get to see if a cell can attach to 1, 2, 3, or no antibodies at all. As long as you have your coordinates set in place, you can develop as many as 40 antibodies and get images and then look, and then the computer can look at them and tell you whether you've seen multi-staining or no staining or 2 staining or whatever. So now if we apply known markers to HSCs, OBs, MKs, and OMs, we can put as many as we want, and we can make those measurements that we are thinking of making happen. So we identify HSC with a lineage cocktail, which is made of these 5 antibodies, along with SCA1, CD117, CD150. And you will see as I present to you some of the data we have, that these are very, very rare cells. Mind you, we did not use in this combination what we would call in flow cytometry a dump channel. In other words, we did not stain for these 5 markers with the same fluorochrome, but each has its own fluorochrome. And so we can see a B220 cell separate from a GR1 cell and so on. The osteoblasts, we went back to what we had been doing before and basically used, oh, I have a typo here. This shouldn't be there. A lineage negative, SCA1 negative, CD166 positive. And these are the labels that we use for megakaryocytes and the OMs. So we have 13 antibodies that we use. We apply them in 7 cycles. By the way, I say we as if I have done the work. I haven't done any of this. So we had to verify that we made this, that we have the antibodies to stain the cells with. Unfortunately, the antibodies are not commercially available. So all these 8 out of 13 were available as purified antibodies. They were not tied or conjugated to oligos before. So we had, or in this case, Sonali had to conjugate the antibodies, GR1F or ATC kit and so on, to the barcodes and to verify that they are attached. And you can see that, for example, c-kit, when it's purified, has 1 band. When it is conjugated, has multiple bands because multiple numbers of oligos bind to the antibody, and so it appears it is heavier and is bound to multiple numbers of barcodes. Now we need to verify this along with a positive tissue, because we have to identify, or we have to verify that the chemical modification of the antibody did not affect its ability to bind to the cognate antigen, which we can do, for example, on the spleen. We chose the spleen as our positive tissue. And we have to show that even if it doesn't bind to bone marrow, it's not because it is not functional or it's not binding anymore because of a problem, but it is not binding because it cannot find its cognate antigen. So in other words, we see a positive on the spleen, but we don't see a positive on the bone marrow. That's fine. That's what it should, that's how some of the antibodies work. Okay, so this is some validation work. You can see that we deal with the spleen and the bone marrow. And by the way, the Codex the slide that we prepare for the analysis has to have both the spleen and the bone marrow, because in every single staining, we have to show that the antibody binds to spleen tissue, as I'm showing you here, and it binds or does not bind to bone marrow tissue. In this case, it does. So you can see, for example, for F480, this is the binding here on the spleen cells. This is the binding on the bone marrow. The validation can all be done in the same color because it doesn't matter. We're not trying to see co-expression. We're seeing the expression of an antibody, for example, CD41 on the spleen cells as well as the bone marrow cells. So we have the 13 antibodies that we want to deal with. How are we going to do this? So the other thing we have to do is we cannot, with the human eye, read the data. We have to rely on a software called HALO, H-A-L-O, that we depend on to make the measurements that we want to do. But we have to tell HALO what we are looking at. So here's a section of femur in which these are bone marrow cells. They are contained within the yellow compartment of the slide. This is bone here, and it's contained within the green compartment of the slide. We have to identify this region and that region for HALO in order to be able to work within these regions and make the measurements that we want to make. One thing I forgot to tell you, which is very, very important. We have to stain with DAPI. DAPI is a nuclear stain. And sorry, I didn't do this before, because you have to identify a cell, because HALO will ignore any staining that you see that is not associated with DAPI. So if the cell is DAPI positive, HALO would go on to analyze it for other markers. If it is DAPI negative, it's going to ignore it. So let me show you how we can identify some cells. So this is the bone marrow. This is an image where DAPI is turned off. By the way, we can turn on and off any color that you want, that you are looking at. So DAPI is turned off here. DAPI is turned on the left side. And you can see, based on these parameters here, that the 45 plus, 41 plus multinucleated cell was identified here and there, so on. And so these are cells that are defined as megakaryocytes. These are cells that are defined by macrophages. Again, this color can be any color you want to. I mean, they are not necessarily the colors of the fluorochromes on which 45 and F480 or with which F480 and 45 were associated. And these are the macrophages. Now, remember, we have to deal with osteomacs. So these are physically close to the endosteum. They are not, and if this is the bone marrow, again, this is with DAPI turned off, DAPI turned on. They cannot be somewhere here. Now we have to identify whether this is the bar scale, so it's 20 microns. So this is probably, let's say, 35 micron away from the endosteum. We have to tell HALO to look for cells that are positive for macrophage markers that are only within, let's say, 35 microns of the green, the green area, or within 35 microns of the green area we're looking at. It cannot be too far out because we will not consider it as an osteomac. here's the definition of osteoblast. We are now looking at the endosteum, so we're no longer looking in the bone marrow region. We are looking in the bone region, and these are the cells that are identified by the combination of these colors that are obese. Now, this is where it gets a little bit complicated, and we cannot do it by the human eye. This is again the bone marrow here. This area is probably sloughing off. So again, we have to tell Halo to ignore it. So we are only looking at this area. This is without DAPI turned on. What we did here in order to convince ourselves that we are looking at LSK cells that are CD150 is that we hypothesize that LSK are going to be present in larger numbers than LSK 150. So we can identify 3 cells that are LSK 1, 2, 3. And sorry, the magnification is not higher. We have to look in large areas to find them. But here's the interesting point. Now we're looking at LSK CD150, and we can only find one. So this is, I'll go back, this is the same area. We went from 3 cells down to 1, and it is this one. You can see a square around it. It's because Halo puts a square around the cell of interest. So this is the only LSK cell that is what CD150 that we can find. Again, look at where the endosteum is very, very close. So we are confident that this is an LSK cell that is also CD150. It is sitting next to the endosteum. So this is the bone, and this is the bone marrow.
Can I answer a question of that image for just a second?
Certainly.
So on the left-hand panel, this is me, maybe it's my vision. I see blue and I see white dots.
Yes.
And then in the middle panel, I see the box.
So DAPI was turned off here.
Right. So the white dots are the SCA1 stain and c-KIT staining. Is that?
We don't know, Diane. It could be any. This, for example, this cell here, we don't know what it is, but it could be lit up by any of these markers, but not by these 2. It could be lit by this one, but not by something else. Or it can be 2 of them, but not by the 3rd. So only Halo would be able to combine all these markers or all these colors, that if you want to say it that way, and then identify this as a cell and add CD150 to it. So that's why it's so difficult to do it by the human eye. But these are real cells. They are lit for being positive for something. We don't know what it is.
Okay, and then my next question, just briefly, a lot of those markers that you're using are plasma membrane markers.
Right.
Right. And so the DAPI staining is a nuclear staining. And so you're seeing aggregates on the plasma membrane of these things. Is that what I'm looking at?
Well, we should be able to see. We shouldn't see a punctate staining of the membrane. It should also all be. It will be all around the membrane, along with DAPI staining.
Eddie, let me chime in here. So
chime in.
Sorry, Ken. I'm just trying to understand the image.
No, it's hard. I get that. I think about these things all the time. So it's anyway. The first thing that the Codex software does is identifies nuclei.
Okay.
And the next thing it does is it radiates out from each nucleus to identify the boundaries of the cells.
Okay.
Okay, and then the Halo software then does cytometry on those cells as they've been identified. And so to the degree that there's something on the plasma membrane of a cell, ideally what you're hoping is that the software is doing a good enough job to find the boundaries of the cell that correlate with that nucleus. So that's how you have a probe that's not in the nucleus and ends up getting associated with a particular nucleus.
Okay, okay.
So in other words,
It's just not inherently obvious in a typical like immunofluorescent staining that you might look at.
so in other words, what Ken is trying to say, if the association between a clear nucleus identified by DAPI and a clear membrane staining identified by anything is not made, the software would ignore it, because these could be, and I'm not sure unless we magnify this image here and look at them, could be non-specific staining of something.
Okay, that's exactly was kind of, that gets to what my question was going at. I was trying to
Okay,
understand that difference.
But most of what you are seeing is specific because the software identified it with DAPI, and you can see it.
So Eddie, if I'm sorry, I haven't seen this before, so this is embarrassing. Do I understand you correctly that the white cells in the middle panel, or the white cells for that matter in the left panel, that those are cells that Halo identified as being Lin minus, but SCA1 and c-KIT positive?
For most part, yes, yes. They have a combination of some staining,
Right.
but they don't qualify for all the parameters that we told the Halo to look for.
But it's satisfied enough of them to identify them as LSK cells.
Exactly.
Okay. Yeah, and that's the kind of thing that, and I think everybody needs to appreciate the fact that we're talking about these gigantic image volumes that can't be manually annotated. You can't go through cell by cell by cell and identify, okay, it has this, but not this, it has this, but not this. You depend on the automated image analysis to find these things for you. And at the end of the day, one of the things that we have to do going forward is we have to go through, and like Eddie said, we need to look at these things very closely and make sure the segmentation is working properly and the attributions are working properly and all that stuff. So this is why it's still, the image analysis is a work in progress.
And do you do single antibody staining, sort of just to confirm that's the same? I mean, I'm kind of, okay.
That's the one that was done, Diane, in the validation process.
Perfect.
Every antibody was validated. And that's when we do single color staining. But
Great.
in the Codex approach, all the antibodies are put together at the same time.
Great. Thank you.
One quick thing that I'm still a little confused about on this. Why on the previous slide, I thought you said only 3 of them were LSKs. But then on the next slide, it looks like there's a lot more than that. Or why were those 3 LSKs, but the other white cells are not?
I went back to the previous slide. This is the one you're talking about, right? Okay. So our rationale was that LSK cell, since CD150 is a subset of LSK, there should be more LSK than there are LSK CD150, correct? Okay, so here we were able to identify 3 LSK cells, 1, 2, 3. Now the cell looks, it has a square around it. That's because the Halo does that. Okay, but these all came from these, let's say 200 cells here. So 200 of them are something that that they fulfill one of these, one or more, or combination of these parameters, but not all of them.
Okay.
So we have 3. Now, we then asked, out of these 3, 1, 2, 3, which is an LSK that is also CD150, and out came this one.
Okay, gotcha.
So we felt a little bit more comfortable that it is working as we expected. That doesn't mean anything. So now we haven't done it, but we are 90% there that we can do these kinds of measurements. We just have to stain another, or maybe this one, the section that we have, and make those measurements. And then see if we have the tissues of the old prepared and we have tissues of the young prepared. So this is all I have for you. And so we have those sections now prepared. The question is, is the hypothesis correct? Is A in the young smaller than A in the old? We don't know that. Or is it numbers? Is it something else? I don't know. We don't know how many we should measure, because as you can see, the LSK 150 is a very rare cell population, especially if we look in the diaphysis of the bone. We should be probably looking only in the epiphysis. We still have some challenges that Sonali and Connor are working on, such as autofluorescence. I'm just going to say something about the process. They being Sonali and Connor, found that photobleaching is probably the best approach to autofluorescence. Now, photobleaching is basically exposing the tissue that we want to stain to UV light. This process has to go all over the slide, all over the section that we are trying to stain. This is a process that can take 27 hours on the machine just to photo bleach the section that we are going to stain and look at. Otherwise, autofluorescence is everywhere and we cannot read anything that we stain the cells with. So it is a time-consuming, the volume of data that is collected is huge. And so we have to spend a lot of time processing those images to reach a point where we can show something that we are confident about, because it takes a long time just to process the data, basically. So it's a time-consuming effort, and we're getting there. We're not there yet, but we're moving forward. I think this is all I have. Yes. Questions?
Actually, I have one thing I'd like to ask the group, for that matter. The autofluorescence has been, I think, so far the biggest challenge that we've found. And there are a variety of commercially available autofluorescence quenchers. And there are some fluorescence quenchers that are incredibly toxic and you just don't want to be around. Lithium borohydride, for example, is one of the things that's out there. The problem that we have thus far is that the mechanism by which they quench autofluorescence is the same mechanism by which they quench fluorescence of the stuff you care about.
They quench life.
Pardon me?
They quench life.
Yeah, and so we're still, photo bleaching right now is the best option, but unfortunately it is, like Eddie said, it's very slow. And in fact, the Codex system is something that runs very slowly. I mean, the whole process is one of solution changes and extended imaging at relatively high resolution. And so we have experiments, Codex experiments, that take place over the course of a weekend. So the problem is that, for one study. And so what that means is that your feedback loop is incredibly extended. It takes us a while to find out that something didn't work, for example. So we're working very diligently on that. We, again, Eddie and I don't do anything. This is all Connor and Sonali. But yeah, I would be interested in hearing from other people in the group as to how they've accommodated autofluorescence, which we find to be really egregious.
Anybody is welcome to unmute themselves. Are you finding the autofluorescence in a particular cell population, or is it everywhere?
It's everywhere, and it's across the whole spectrum. It's like trying to find the moon in front of the sun.
See what happens, Diane, when I mean, Sonali, Connor, Ken, and I meet regularly to try and figure out how to move forward with this. And in several meetings, we've had images with autofluorescence. You can't see anything. The whole slide is red. You can't see anything with it. I mean, and it's a wide range of wavelength that the autofluorescence comes across, and you cannot see anything. So, but the bottom line is that will interest those who are still around listening to this and want to even check it out on the website. Once we get this working, it is going to be a resource and on the IU flow and tissue cytometry core for anyone that wants to use it. So please, if you have an application, ask us or try to start early, and we'll see if we can help you. But this is why we're developing it.
It's a great technology. I don't have any good answers for the photo bleaching. I'm sure you guys have tried all of the things that I would suggest about. Are you doing a constant UV at a low intensity, or is it like bursts, or exactly how is that photo bleaching done with the UV?
Connor, can you chime in?
Yes, hi. We do a constant lamp quenching, autofluorescent quenching with the photo bleaching with the lamp. So it's just the on-stage lamp with the microscope that we use to acquire the images. So we're actually baking every region that we acquire for about 30 seconds.
Yeah.
But the system isn't a confocal, so the tissue is getting generally bathed in light for, as Eddie said, about 24 hours, 24 to 27 hours.
So Diane, one of the quenchers that Sonali tried to use was forgot what it is.
TrueView?
Yes, thank you. True Black. What happened with it is that True Black itself was so dense. I'm using the word dense only because it describes what happened, that it basically covered the fluorescence that was emitted from the fluorochromes that we were trying to use. It was basically covering everything that we couldn't see it. So we had to abandon that kind of a quencher.
Yeah, I have not used that. What I do know is I think a lot of the fluorescence, the autofluorescence is coming from lysosomes, but your tissues are fixed. So I'm assuming you're trying to think about what you could do to quench that fluorescence in the lysosome.
I mean, if you think of something, it would be really helpful. We can try it and it would be really helpful.
I'll kind of ponder on that a little bit.
So
I mean,
one thing,
I'm not convinced that it's lysosomes, to be honest. I've seen a lot of autofluorescence from lipofuscin, typically is what it is in lysosomes of proximal tubule. And it has a really distinctive pattern to it.
Okay.
Lysosomes is punctate, it's fascicular and that kind of thing. And
Right.
yeah, and that we might be able to deal with, but this is a much broader. Connor, can you talk about?
yeah, I think that it's so we are using formalin fixed paraffin embedded tissue.
Yeah.
So I think that it might be coming from the paraffin. So if there's even some suggestions for how we might do tissue recovery and antigen retrieval from the paraffin, if we can more completely remove the paraffin, I think that'll go a long way to getting rid of the autofluorescent source.
Right. I mean, there's actually a reasonably rich literature in what you need to do in order to get rid of the autofluorescence that derives from paraffin. And it has to do with how getting rid of all of it. And this is something that we've been at it for a while, but not that long. So I think that we can explore that as well.
It seems like we need some sort of blog out there for this kind of communication besides these talk times where people can be sharing ideas about that. There's probably something out there. I just am not aware.
It's one of those things that I think once you solve it, you stop talking about it, and if you don't solve it, you definitely don't talk about it. So the problem is we've run down a lot of blind alleys that other people could have said, oh yeah, that doesn't work, but that's never published, so.
The questions about what should get out there for all of us, that we don't spend the same time inventing the wheel. Yeah.
Sort of.
Anyway, that great talk, Eddie. Any other questions that anyone has? Just for those who are watching and know of anybody who might be interested in hearing this talk, we will post the recording on the website within the next couple of days. And we also, in that imaging microscopy seminar series, will have more on this from Dr. Ken Dunn that happens in September. So stay tuned.
And actually, before everybody bails, if everybody hasn't already bailed, I want to point out that bone marrow has been easily the most challenging thing that we worked with. We've done Codex on all kinds of tissues, including lymph node, spleen, all kinds of tissues that might be of interest to you, and had terrific luck. So yeah, we're imaging upwards of 20 different probes in a lot of different tissues, and not encountering the issues that we've got with the bone marrow. So to the degree that you're interested in imaging tissues other than the bone marrow, it's a much more encouraging story. And I guess I'll be presenting that encouraging story in like October or something.
It's September 12th.
September 12th. I should mark that down probably.
Only bone marrow counts, Ken.
The only bone marrow counts.
Dang, as usual. Okay, terrific talk, Eddie. Thank you so much. And thank you to those attendees. If you have
Thank
other
you all.
questions, I encourage everybody to reach out to each other, and you're welcome to reach out to me if you have other questions, or if you need help connecting with an individual who might help you with imaging microscopy at all. And it looks like it's going to be a great core when the details do get worked out.
Thank you, Diane.
Thank you, and everybody take care.
Okay.